All right. Well, good afternoon, everybody. Welcome to Samaritans International Health Forum. As always, we're just delighted to have you join us this afternoon. It's a beautiful sunny day here in Boone, North Carolina, with hints of spring. So we're excited, and again, excited to have you with us. If you haven't joined us before, just utilize your chat box. We encourage you to utilize that, especially towards the end. We will dedicate some time to Q&A, which you can use the chat box to accomplish that. And before I introduce the speaker, I'm going to open us up in a word of prayer. So let us bow our heads. Dear Heavenly Father, we just thank you for this day. We thank you, God, just for the opportunity to broadcast Samaritans International Health Forum, that we may know more to serve you better. And I just pray, Lord, for Dr. McGinnis today, that you would just speak through her and just bless this hour. And we just ask this in Jesus' name. Amen. Okay, well, it's just a real pleasure for me to introduce Dr. Terry McGinnis to you today. She's going to be speaking about a COVID-19 update, vaccines, immunity, and variants. Of course, a very relevant topic. Terry, currently, she serves as the president of a consulting firm, Bluethorn Incorporated, and she's also the chief medical officer for Arene, which is an artificial intelligence-driven technology company that's really devoted to optimizing pharmaceuticals. Terry really serves as a strategy consultant in value-based care, and really she's been successfully positioning organizations to take on risk in the healthcare industry. Terry really has a broad perspective and has served executive roles in many segments of the industry. I'll just name a couple in the diagnostic industry with LabCorp, and pharmaceutical with GlaxoSmithKline. Dr. McGinnis, of course, is a physician first at heart. She served with the United States Air Force as a flight surgeon during Operation Desert Storm and Shield. She also, in her earlier in her career, she supported 38 space shuttle launches and landings before she transitioned to civilian and industry practice. Dr. McGinnis earned her medical degree from Wake Forest University here in North Carolina, and then she went on to complete a residency in occupational medicine, and then received her MPH from the University of Oklahoma. Dr. McGinnis is board certified in preventative and occupational medicine and is a certified physician executive. So Dr. McGinnis, you are a busy person, and I believe you said you're coming to us today from Arizona. So we just really appreciate your time and your words today with regard to the COVID-19 update.
Perfect. Lance, thank you so much. And I just first want to thank you and Samaritans and everybody that's listening to this for all the work that you're doing. And you literally do God's work, and you do it in some of you are doing it in some incredible places around the world. So it's just my pleasure to be able to be with you today. So if we can have the slides, we can get started. So, it's kind of interesting. I've been giving talks, and it seems like I have to update them literally daily. So this morning I spent a little time with some of the statistics you're going to see. And I always say, it's almost like the questions are the same, but the answers keep changing. So hopefully we're all learning together, and I'll share a few things, and then we'll have time for Q&A. So as Lance said, we're going to look at sort of the vaccines that are out there today, what we know about immunity, and some of the troubling issues we're seeing with variants, and also what I think is the silver lining. I think God always gives us a silver lining, and I think I'm going to share what that looks like in the midst of this pandemic. Next slide. So actually, New York Times has done a great job in terms of really tracking both vaccines that are being developed around the world and a lot of the good statistics. So you'll see some of my references here, but just as of yesterday, there are actually 75 vaccines in clinical trials and development, and 21 now have reached what we call the final stages of testing. And Another 78 are actually under active investigation in animals. So one of the things that this has done is just really set a spark to vaccine development and innovation. You see now there's 6 that are approved for full use throughout the world and 6 that are authorized. Next slide. So if we look at some of the leading vaccines out there, what we see is that I think many of us have heard about the messenger RNA vaccines, both the Pfizer, BioNTech, and also the Moderna. They've really been proven to be close to 100% effective against severe disease. And we've seen varying things like 94 to 96% against actual infection. The interesting thing we've seen about that, though, is the cold storage issues, which have really limited a lot of that use to developed countries and not really use in developing countries that really couldn't do a lot of the cold chain. However, there is one in the pipeline, CureVac, that is actually stable at close to basically refrigeration temperatures at 44 degrees. I should have put Fahrenheit there for 3 months. We also have, you've heard a lot about the adenovirus vaccines or adenovector, and Johnson & Johnson just got approved here. Again, it's being used also in multiple other countries. And even though the efficacy against disease itself, symptomatic disease, is less, we have about 100% so far. I hate to ever say anything's 100%, but our NIH director used it. I'll say 99% against death and severe disease. And the one thing I want to point out there, it is a 1-dose regimen. They're also looking at a potential 2-dose and the efficacy around that. But J&J has been approved for emergency use in the US as a 1-shot vaccine. And then in other countries, we have the Sputnik 5 that showed very good efficacy too. Obviously, that came out of Russia. It's being used in several other countries too under emergency use. And then we've also heard about the Oxford AstraZeneca vaccine. That's a 2-dose vaccine. We're seeing it used in the UK and again, some other countries. We also saw, though, that it was stopped. That was a vaccine that actually was halted in South Africa because they really thought that the efficacy was not quite as great against the South African variant. And then we have, so just to note right now, the only 1-shot vaccine that we have is approved as Johnson & Johnson. Might be important for later test material. Next, you see an interesting vaccine that's getting close to approval, and that's Novavax. And it's actually an interesting nanoparticle protein vaccine. And again, it's showing pretty good efficacy against most variants, about the 50% in terms of any kind of disease, but at least it looks like it's against severe disease, it's much higher for the South African variant. And then you have, there's 2 out of China that's used right now in China. Actually, I think they just hit 3. So the Sinopharm, the Sinovac, and the CanSino. And that's an example of inactivated viruses. And I put another one up here that's actually emergency use in India because it's very interesting. It's being used as a nasal spray. So again, that obviously, when you're not having to inject, that makes it much easier for distribution in more rural and developing nations. Next slide. So if we look at how quickly immunizations have gone up, it's just incredible. So just a couple of weeks ago, we were at 216 million, and literally another 100 million vaccines have been given in about that 2-week time to get us up to 316 million worldwide. And the thought is that, if it's just the developed countries that get vaccinated, we're going to really continue to have a problem with variants and spread until we get more of these vaccines out globally. Next slide. And this is what you see. We do see that uneven distribution. I think some of the places where people here in Samaritan's Purse are very familiar with, especially you see Africa, has really not had vaccines available. But again, I think that is a lot of work internationally, and I'm going to share that. I'm going to share that here shortly. In terms of the countries who have actually had the most shots in arms, Israel is leading, and they just came out with a paper really looking at the efficacy. Basically, all the vaccine given in Israel is the Pfizer vaccine, so they don't have a mixture of vaccines. But what they're finding is they actually have some of the UK variants and other variants, and they're finding very good efficacy in terms of, again, against severe disease and hospitalizations and deaths. So good news there coming out of Israel. And you can see other areas like Serbia and UAE that are getting also a lot of shots in arms, followed by the UK and United States. Next slide. So I just want to spend a little bit of time then on what's happening internationally. And COVAX is really the initiative that's working on global equitable access to COVID-19 vaccines. And the G7 countries have already committed $4.7 billion collectively to this effort. Some of the foundations like Bill and Melinda Gates have actually put a lot of money into this. And the purpose of this is to actually the goal is to have fair distribution of vaccines in developing countries. And that goal is actually to have 2 billion doses distributed by the end of 2021. And they're doing that several ways. So in addition to the World Health Organization, you have CEPA and Gavi. I'm probably mispronouncing those, or at least the acronyms. But CEPA is really involved. It's called the Coalition for Epidemic Preparedness Innovations, and they are really behind the research and development, investing with multiple different companies to try to get various vaccines in the pipeline that are more friendly for developing worlds. And whereas the Gavi is really the procurement side. So Ghana actually just started immunizations March 1st. So again, that's a glimpse of what's happening in terms of global, trying to be more globally equitable in terms of vaccine distribution. And I, again, have a website there if any of you want to continue to kind of track that. Next slide. Okay, so let's look just a little deeper at Pfizer and Moderna. So both Pfizer and Moderna are 2 shots. You see the Pfizer being 3 weeks apart, and you also, the Moderna is 4 weeks apart. Both of these are multi-dose vials. They're very, I guess, somewhat fragile. So for example, once Pfizer's diluted, you can't shake it. You just have to invert it. With Moderna, you swirl it. So again, some of the challenges, including cold storage, even though Pfizer has gotten a little better now, it's not as cold a storage chain now. They just got approved for a little bit more refrigeration, normal refrigeration temperatures. That has been part of the issue. And if you look at then what's happened to patients, we've literally had millions and millions of doses now of these 2 vaccines throughout the US. We really want to observe patients for 15 to 30 minutes due to the immediate reactions, including anaphylaxis. And I'll talk a little bit about that in a second. There's a lot of good information that the Centers for Disease Control here in the US has put out under the Emergency Use Act with the fact sheets for both recipients and for those that are vaccinating. And in the US and worldwide, we're seeing a call for more vaccinators for things like those physicians and nurses that actually have expired licenses within the last 5 years. They've been recruited as vaccinators. And obviously we have pharmacists here in the US, but beginning to add, even thinking about bringing dentists in, vets, advanced EMTs, and medical and nursing students. So if you think about what's happened with the vaccine, at first it was real supply issue in terms of just getting the vaccine produced, getting it out. But basically, what we're seeing now is that we've got to get the supplies catching up, but we've got to get more shots in arms. Next slide. So what we're seeing in patients, and as I was sharing with Lance before this, that's one of my volunteer efforts. I think that I've volunteered now for seven shot clinics, maybe 6 or 7, and so have actually given Pfizer and Moderna quite a bit. And Johnson & Johnson is just getting out now, so I get to volunteer tomorrow to give Johnson & Johnson vaccine. But I want just to make sure everybody understands sort of what to tell patients and what to look out for. So local reactions are very common with both of these vaccines. And I will say what I'm hearing, again, I don't have experience, is really the same. You can think about the same things with Johnson & Johnson, even though it's not a messenger RNA vaccine. They're seeing that, again, local reactions are common, like injection site pain, but these systemic reactions are also pretty common, especially the first through second days post-vaccine, could be into the third day. And that includes systemic things like headache, fatigue, fever, muscle aches are most common. I'd say about half the people really say they don't have hardly anything, maybe a little arm pain. The other half are really having some fairly significant systemic reactions. And again, when I say significant, like myself, I had 101.4 fever and kind of body aches for a day and a half with my Pfizer vaccines. Again, it's more common after the second dose in younger people, and also with a previous history of COVID-19 infection. So if you think about it, if you've been infected before, that primer dose, which is usually the first dose, really is acting like a booster. And so we're seeing a lot more of the systemic reactions, I'd say, in people that have been previously affected. And then the lymphadenopathy is something that has come up, and that's just a caution because we're radiologists, with mammograms, they're looking at this, they're seeing lymphadenopathy, and it's just been something that we've had to make sure that we're not saying, hey, not mixing this up with breast cancer. So what are contraindications to the vaccine? Really, they're very, very few. It's really a history or allergy to PEG or polysorbate. The same with Johnson & Johnson. It's polysorbate. It has that in the vaccine mix, and that's really where that's commonly found is in bowel preps. And patients usually know if they've been allergic to this, but that's one thing I'll ask: have you ever had trouble with bowel preps in terms of a anaphylactic type reaction? The other thing is, if you've had a history of an anaphylaxis to vaccine, it is not, if you have a history of anaphylaxis to these vaccines. So if you've had the Pfizer or if you've had the Moderna and you had anaphylaxis to that, then that's a contraindication to the vaccine. Also, you should delay 90 days if you've had a history of actually antibody treatment or convalescent plasma due to COVID, due to having COVID. Because again, you're going to have that circulating antibodies, and that's just basically going to bind the vaccine, and you're not going to get the active, your body's not going to then have a chance to actively create the antibodies and T-cell response. So it's better to delay. So I think it's very important to note the real 2 contraindications, there's only 2, is to components of the vaccine or anaphylaxis to the vaccine, not to other vaccines, to those vaccines. That might be important later. And then commonly people can take acetaminophen or Tylenol or ibuprofen for symptoms, and that's usually enough to take care of some of the local and systemic reactions. Next slide. So again, we've heard a lot about this, patients ask about it, but anaphylaxis is very rare. So we're talking about between 2 and 6 per million. If you kind of look at both Pfizer, or 2 to 5 per million. And all of these all that I've seen to date have been successfully treated. I haven't seen the latest, but through January 18th, there had been no deaths from this. But it's very interesting to note who's having these anaphylactic reactions, even though they're very, very rare. And if you look at the data here, it actually appears to be people under age 63. So nobody in the early data was over age 63. And it tends to be mostly women. So younger women tend to be the ones that are getting this. And again, that might be an important kind of thing to note. And so again, when you're talking to patients about this, that sounds scary. That's why actually, if they have any history to anaphylaxis, to bee stings or penicillin or other things like that, we actually ask them to wait 30 minutes because most of these also occur, most of these reactions have early onset, really within 15 minutes. And you can see that the vast majority, or 89%, occur within 30 minutes. So that's why it's very typical, and that's a recommendation for people just to wait and make sure they're not going to experience one of these. Also, most of those are 80% are after the first dose. And again, if you have it in first dose, it's now a contraindication to have that second dose. So that's where you see, again, most of them. Notice here too, what I haven't mentioned. Things like pregnancy are not a contraindication to these vaccines. Being immunocompromised is not a contraindication to these vaccines. So again, people can talk to their doctors about it, but those are not, in terms of labeling of the vaccine or recommendations, those are not contraindications. Next slide. Okay, as I mentioned, the J&J vaccine has just been approved in the US. We're seeing it also in other countries. As I mentioned, it's a single dose. It's the only shot right now that is approved for a single dose, even though we see other countries like the UK doing single doses, then in some points waiting much longer, like up to 13 weeks. To give a second dose if they are given second doses. But when you look at how the clinical trials ran and what is currently approved, J&J is the only vaccine right now that's approved for single dose use. It's also been touted that you can use the normal refrigeration temperatures, which is great, but there's this little caveat that is very interesting, that after you actually open the vial, and these are 5 multi-dose vials. If you don't store it then under refrigeration, you only have 2 hours before you have to discard it. And being in some of these mass vaccination campaigns that I've done, we vaccinated over 1,150 people one day. That gets a little problematic when people are drawing them up and we're kind of looking at those timelines to put shots in arms, where Moderna and Pfizer actually, after you either dilute Pfizer, you open up the multi-vial for Moderna, you actually have up to 6 hours at room temperature use. So just something to note there, in terms of that makes Johnson & Johnson, in my mind, a little less friendly in terms of mass vaccination efforts. Next slide. Okay, I'm going to that's kind of everything I have on the vaccines themselves. So you've heard a lot about mutations. So I thought I would take us back to medical school here to understand just a little bit about this. I'm not going to go into too much detail, but if you begin to look at this, is, messenger RNA is basically ribose nucleic acids there are 4 of them. You remember cytosine, guanine, adenine, and uracil. So that's the CGAU. And basically what happens is, when a mutation, there's various different mutations that can occur, but one thing could be a point mutation, and that's where one of these bases is actually changed out for another base. So in this example, you actually see where it says missense mutation there. You see the GC, and the upper one, it was GG. Now it's turned to GCU. And what that did was actually then change out the coding of which protein, which amino acid is going to make up the protein. So it actually switched out from the top glycine, in that case, to alanine. And what that does is, if you go to the right, you've got now a change in that one amino acid at the end of the protein, so versus the original one. And according to what that change is, sometimes very like-minded or similar amino acids are switched out. They might be just the kind of small polar ones, but if you might have a phenylalanine, which is a big aromatic, it might really change the protein structure. And then what that means is these proteins, then if we're thinking about things like the spike protein, which is the most, the one we're really concerned about, because that's what combines to our ACE receptors, that conformational change of the protein really can change a lot of things about the behavior of the virus. Things like transmissibility, how closely it binds actually to that ACE receptor. Is it tighter? Did it change the configuration to such an extent now that your antibodies, if you've had COVID or if you've had the vaccine, that now those antibodies don't recognize it because it's changed so much? So basically, sometimes we have mutations that really don't make any difference at all. So you might have the same amino acid. That's called a silent mutation. You see that example here in the first bullet of a GGU going to a GGC and a GGA. So changing that last member of that codon, which is the 3 sequences that go into the amino acid, all of those still equal glycine. You don't see any change in the ultimate protein structure, but some actually make these, some actually make a change in the amino acid itself. And some mutations will actually, like a frame shift or a nonsense code, will actually then stop the protein altogether. And what you're going to have is the lack of viral replication, and then you're not going to have viable virus. So there's a lot of things that can just stop the virus too. So that's just a quick sort of, as people talk about variants and mutations, I think it's kind of smart for us physicians to at least maybe go back and review a little bit of biology and know what's happening. Next slide. And this is just one of those tables. Again, I got some of this from Khan Academy, so you guys can go back to look at your MCATs and college. But again, part of this is that translation then of the RNA to the amino acid sequence. And as I mentioned, these cute little tables here that show if you start out with G in the center here, then go out to the next layer, there's the C, and then go to the next layer, C, that equals alanine. And so that's how the basically how that coding works to figure out what the amino acid is and then what the ultimate proteins are. The reality here, though, so that's enough of the biology lesson. The reality here is, though, that mutations are inevitable and they spread. And that's actually why we are all different. God made us that way. So while we have different hair color and different eye colors and different characteristics, some of us are tall, all of that's because of our genetic variability. So that's good. And what we see is viruses and bacteria, though, obey those same laws. They continue to mutate and they pick up different characteristics. And sometimes those basically kill the organism, but sometimes it makes it really more transmissible, or it might make it bind more, or it could make it more severe. So, or to really elude then or evade the defenses we've already set up, like if we've already been vaccinated against a certain sequence in a certain way the spike protein looks, in this case, the receptor binding domains, which a lot of these vaccines are going after. If that looks too different, then it's like our body thinks that our body doesn't recognize it and it doesn't react to it. And we have to, that's when things like reinfections occur. Next slide. So just looking at then some of the key mutations, because we're hearing a lot about them. And again, not going to go in too much depth here, but this is the B1351, the South African variant you've heard about. It has 3 kind of major mutations that are problematic. And as you can see here to the right, this is the spike protein, and you see the 3 subunits. You almost see 3 spikes there. It has 3 subunits. And then the receptor binding area, you see where some of these, like here's the N501Y, you can see if it shows up once, because there's 3 subunits, it shows up 3 times. So it's kind of like the subunits make 3. So the E484K which is really one of concern now that we're hearing about in terms of evading the antibody responses that people have either to earlier wild infection or to vaccines. That conformational change in the spike protein has been one of the ones of concern. Next slide. And this is just one more example. This is the L452R mutation, which is again one that's been seen. That was first recognized spreading in California. A concern there is it does this. It exchanges arginine to replace leucine at the receptor binding domain. Again, one amino acid change can really make a difference. And what they're saying or seeing with this, it creates a much stronger attachment of the virus to human cells because it actually grabs tighter to the ACE2 receptor. And again, a concern about avoiding neutralizing antibodies. So the convalescent plasma we're using, the antibodies that have been developed to treat severe cases, they're not working as well with this mutation. Next slide. Okay. So here's some of them that you've seen. The B117 is the one that emerged out of Britain. You can see it really has spread rapidly to, it's in 33 states in the US, 70 countries. The South African was spread. It's also in the US and in many countries. And the P1 was the Brazilian. What you might have seen recently, I think it came out on the 8th of March, was a study looking at the P1 variant, the one out of Brazil. And the good news there is both Pfizer and Moderna vaccines seem to have still pretty good efficacy against that mutation. So again, as I mentioned, the major issues, it could make more efficient viral transmission, could mean more severe with certain mutations, greater morbidity and mortality, and vaccine escape or ineffective, and the antibody treatment ineffective. And I will say with the Brazil one, they saw, they began to see reinfections in a town that had really been hit hard with COVID earlier. And that was due to the P1, that the people just did not have good natural immunity after the first one with a change in the P1. Next slide. Okay. Interesting. One of the things that I'm going to, so why is this important? I guess that's the question. Besides the obvious that I've just shared in terms of, the vaccine in being ineffective, then what does that mean for us going forward? And I think what it means is, one, the good news is all the major vaccine developers are actually now, quote, changing their formulas. So if it's messenger RNA, they're actually making changes to the messenger RNA to account for these mutations and variants. So all of the major vaccines or vaccine producers are not just keeping with, quote, the wild type, the original Wuhan virus that came out, but actually modifying those. And I think that's going to be important. And what that means is just like we're used to taking, we're used to having the flu shot every year because of the variations we see in the hemagglutinin and neuraminidase components of the virus. Now what we're seeing is we're probably going to get boosters. And the question is going to be, when are we going to need boosters? Shots as these variants come out. So again, that's going to be part of our future. We're not going to be, we definitely know we're not going to be one and done with this virus. Next slide. Okay. As I've already mentioned, that risk of illness from COVID-19 has really dropped. This was some of the real world evidence, about 96% who received both shots. So I think that, again, the vaccines have been pretty good overall, and actually much better than any of us expected. Think about, gosh, even 6 months, a year ago. So there has been, as I mentioned, a small risk of reinfection 6 months after previous COVID-19 infection. Some of that is thought to be waning immunity, but also the variants that we've discussed. And so the question then becomes, how can one shot be efficacious enough as we're seeing kind of a UK strategy, or what do we need to do with the vaccines that are 2 shots, and that's what the clinical trials show. Do we need to continue with the Moderna and Pfizer 2 shots, or people that have had previous infection? Will 1 shot be enough for them because they've already had infection and this really increases it? I think those are some of the questions that we're all continuing to grapple with. Next slide. I wanted to share just a few other things because I said there's a silver lining here. And I just wanted to point out this is a vaccine that's still in development. The key things here, it's DNA based. It is a 2-dose vaccine, but again, it requires no frozen shipment, and you can it's stable at room temperature for more than a year. So when you're thinking about developing countries beginning to get vaccines that are stable at room temperatures, not requiring refrigeration is huge. So just remember this, DNA vaccines fall into that category if you happen to see that on later tests. Next slide. So this is the exciting part. I think any time we know that if God closes a door, he opens a window. And what the window I think he's really opened here in the midst of all of this suffering is innovation in vaccine development. And I just want to give you just a quick snippet of what this looks like. So the computational biology, computer-aided dimensional analysis has just been absolutely incredible. So now what you see on the right, and I'm not going to go into it in any depth, but basically what we're able to do now with the computer simulation is being able to show exactly all the changes that might occur. In this case, at the top, there's your spike protein, and you see the receptor binding ridge and sites. They're actually able to look and say, well, what if we did change out this amino acid? What if we did make these changes? So they're actually proactively now, before waiting for a mutation and say, is it going to be bad or good? They're actually being able to do some of this in the lab and say, here's all the possible mutations that could occur. So, and then this is what it would look like. And these are the ones that would make it worse in terms of binding more to the ACE2 receptor or more severe disease. So it's really incredible, the computational biology that's occurring. Next slide. And the other thing, in addition to sort of this ability to look without ever going to the lab in terms of what the amino acid sequences, proteins, and then how to begin to develop vaccines, we're also seeing then how we can actually make vaccines with multiple different receptor binding domains. So in this example, we're seeing a nanoparticle hub. So if you look at this kind of the middle part where you see that mosaic RBD particle vaccine, you actually have on each of the spikes of the hub a different conformation of that spike. So here's all different variants. So you can actually be given a vaccine that's not just 1 or 2 variants. You can actually, if you can imagine, put together these vaccines with multiple variants that are the ones that, again, you've looked at and computer generated, these are going to be the worst ones, tag them, and now you can give people vaccines that have cross-reactive antibody response. So think about it. Perhaps we could have one that really hits all the coronaviruses. Hits MERS, SARS-1, the 4 circulating regular ones, and really hits anything we might think of in terms of the spike protein that would be of concern, and give a vaccine. Again, this is future, but it's happening very quickly. In other words, this has really opened up things like messenger RNA platforms. Even though you've seen some of it with Ebola, it's opened it up really to hit multiple diseases in multiple ways. So I think we're in for a golden age right now around vaccine development. Next slide. So, and this is just, again, another example. As I said, there's this whole idea that we don't even have to go into cells, or we don't even have to use animals, that really we can have fully synthetic protein vaccine constructs. and really it combines both targeting adjuvants, which are those things that sort of wave and say, hey, come get me, so adjuvants are typically used like if you had the shingles vaccine that had a very good adjuvant that sort of said, hey, come look at this body react. It's kind of like kind of having the siren. And so having the right adjuvants that can amplify then the effects directly to the proteins, which could be viral or bacterial, is really making some interesting things, because it's ending up being sort of one single molecule combined. And one example of that, again, early studies, was there's some significant protection against even tuberculosis in mice. So some of the diseases that we think of that have just been beyond us in terms of real vaccine efficacy, I think have the real potential here in the next several years to really make a huge difference, both in developing and developed worlds. Next slide. So these are just, I'm going to stop here soon and open it up for questions. So these are resources. So again, if you get the slide deck, those are just COVAX and some of the other educational resources. I will just put a plug in for this for you physicians and providers. Medcram.com does a great job too. Dr. Roger Seaholt, it's free for the coronavirus series, and he does great updates and he really well documents his thoughts and references in there. So just kind of a shout out for that, in addition to all the ones like COVAX and others that I've shared in CDC. Okay, next slide. All right, so as I've mentioned, I really think there's this COVID silver lining when it comes to vaccine development. And as a preventive medicine specialist, I'm just tickled about this. So if there is some silver lining in this bleak pandemic, I think that's really it. That it's a way that God's going to use to really, really jumpstart vaccines and really prevent a lot of disease and problems in so many different areas. So all of this is making vaccines, I think also they'll be made cheaper and lasting longer, as we know more and more, with more computational biology models, getting rid of a lot of the trial and error that's been done in the past. Next slide. Okay, that's all I have. So we'll just open it up for any questions.
Terry, thank you so much. You did a phenomenal job of taking a very complex topic and simplifying it for folks like myself. So that was presented with great clarity, and I appreciate your expertise on that. We've already got, I think, 4 questions, so a very inquisitive audience here. Don Rumpel said, and I think this is a great question. COVID-19, is it here to stay? Is it like our annual, the influenza season? And what could you say about that?
Yeah, I think it is. But if you think about it, we've got 4 coronaviruses now that circulate as common cold viruses. And even if you look back at 1918 to the flu epidemic there, that avian flu ended up in normal circulation after it sort of went through, and we had a year and a half of then no vaccine, but basically developing herd immunity. So I think it is around, but I think the good news is we're going to have better and better vaccines to really take care of it. So boosters are in our future. Like I said, all the major vaccine producers are already changing their formulas. And I think with some of those computational biology models, they're going to know kind of preemptively what to put in there, even if the virus doesn't change itself.
Right. Yeah, that is amazing. The innovation that's coming out of vaccine development is incredible, and to, as you said, to preemptively prepare vaccines is incredible, and to couple them and address multiple diseases or most anticipated mutations is great. But Terry Laurel Down says, have you had any experience or have you seen any post-vaccine pneumonia? Apparently she has heard of 4 cases.
I really haven't. And I think one of the things just to recognize is that when you have the vaccine, you still aren't developing immunity. You know, it takes about 2 weeks before you start having significant neutralizing antibodies. So a lot of people, you know, they'll get, you know, there are cases of you have the vaccine and, you know, 7 days, 10 days, 12 days later, you've got COVID. And that's because your body just hadn't built up. So I wonder if some of those pneumonia cases might be just, it's, you know, it's coming because of COVID or other causes of pneumonia. But I will tell you the vaccines, these are not live vaccines. You cannot get COVID from these vaccines. And that's across the board with all the ones that are out there now. So that you can't get. What I have seen is some interesting, I mentioned the lymphadenopathy, some hypersensitivity reactions that are occurring not 1 to 3 days, but might be a week later, where you might have a really red indurated arm and lymphadenopathy on that side. And so those have occurred. But otherwise, you know, the typical systemic reactions I mentioned, and also those very, very few anaphylactic reactions, again, in about 2 to 5 or 6 per million people, mostly women, mostly young, are what we see. And you know, I kind of remind people, because people, you know, it's concerned, am I going to have anaphylaxis reaction from this? But in the US, over 1,500 people per million have died of COVID. So, you know, again, having anaphylaxis, which I'm not sure if anybody's died last January, they were all treated. Some ended up in the ICU, but they all recovered.
Right.
You know, these vaccines are very safe.
Yeah. Yeah. Risk benefit ratio tremendously in favor of getting
Absolutely.
the vaccine. Yeah.
Absolutely.
Yeah. Terry, Tim Tusinik asked, this is a great question. If you had your choice of vaccine, which one would you get?
I'll probably have to go with Anthony Fauci's answer, you know, the first one you can get.
That's good.
And I think that's really the case, because some people have asked me already. I'm just going to be interesting, since I'm doing my first clinic tomorrow with Johnson & Johnson, what people are going to say.
Yeah.
Because people have seen the efficacy difference in Johnson & Johnson, but really Johnson & Johnson clinical trials were done in different places. So they did a lot of their early trials, like in South Africa, where they were having the variant come out. Right. Moderna did all their clinical trials in the US. So we didn't have any of those other things popping up. So I think the reality is, get the shots you can, and understand this, you're probably going to need a booster at some point. So I would take any of the shots.
Right. Even the Johnson & Johnson, I think, with severe disease, and as you said, it was tested in some situations, like in South Africa with mutations, it was like up to 85%. So effective for severe disease. So
yeah, actually, they're saying against severe disease and hospitalizations, it's close to 100% too. It's great. So, you know, again, you're still seeing infections. So that efficacy rate are people getting infected, but they're still not getting the very severe disease. Exactly. So again, that's going to change though. As more variants come out, you know, I think we're going to have, you know, there's potential for less efficacy. The other thing is just, again, this is not a CDC recommendation, but one of the things that's being floated, and some countries have considered it, is people that have had COVID. You've had the natural infection. That's like getting the primer dose, right? You know, you've got a lot of things. People that have then one shot after that, their antibodies and reaction just is really greater than a lot of people that have had the 2-shot vaccine, never had the natural infection. So, you know, I've taught people, I said, look, you know, an easy way to do that now is, if you did have a choice, if I was infected before, I'd go get the J&J vaccine.
Yeah, exactly.
That's just my opinion.
Yeah, no, I respect that. Bruce Steffies asked, some patients are afraid of that nucleic acid vaccines will introduce DNA into their cells. And he said, of course, they don't. But what would you say to them about these DNA-based vaccines?
Well, and you do have some DNA vaccines that are coming out, but we don't have them here yet. So the RNA, yeah, so the RNA vaccines, the way the RNA is actually, you've got the lipid layer around these RNA vaccines. So you got the messenger RNA that's in that lipid layer. So that lipid layer basically allows the introduction into the cell of that messenger RNA. That messenger RNA, if you remember that biology slide I showed, then goes to the ribosome. You got translational RNA, and it makes protein. Then it's gone. Your cell kills it. It can't replicate itself to make more. It doesn't replicate new virus. The only thing it does is code for that spike protein. So there's no way that can get in your body and do anything. So I think that's the answer there, is just, a lot of people just kind of don't understand the difference between RNA or DNA and how we have our, we have a lot of mutations all the time, it's just part of it.
I think some of the concern too was that there was potential maybe for autoimmune disease, but what's reassuring is messenger RNA is rapidly degraded, and there's not continuous replication of spike protein. And anyhow, very good question. A lot of, like I said, very inquisitive audience today. Pat Webster says, what do you think accounts for the dramatic drop in cases since January?
Hey, great question. And I think it's multifactorial. First, I think if you look, you look at various places, but we kind of had Thanksgiving where we had gathering, and then you saw an upsurge of stuff. And then you had Christmas and you had people gathering. You had New Year's, you had people gathering. And so you had this upsurge in addition to the fact that you had a lot of cases already out there. And, some countries have been good and, hadn't had hardly any. So it really varies in terms of you're talking about the upswing. But I think that it was more is happened there. The other thing is we tend to have viruses, whether it's humidity, temperature, or whatever, they tend to be worse during the wintertime as we're beginning to get, kind of past that time, warming up a little bit in certain areas. You're seeing kind of that natural kind of decrease. But the other big thing is we're seeing more vaccinations. And so I think as you see that, the slide I showed with the number of people being vaccinated, that's helping. I think the other thing too, to be honest, is a lot of people that were either in jobs that were more risky, or maybe they had riskier behaviors. We've had a lot of people with infection in this country. So, now you're kind of taking out, since reinfections within 6 months are pretty uncommon, you're kind of taking them out of the pool of continuous continuous propagation of that. So I think that the answer is there's multiple factors.
Yeah, exactly. Thank you. Stephen Willing asked, and I think you've already addressed this to some extent. He said some that are claiming deaths from the vaccination, i.e. anti-vaxxers, but he said, I'm highly skeptical. Any credible evidence to date?
No, no. And they really are I think they're doing a really good job in this country and other countries of really looking at any deaths around vaccines. And the reality is this, I think that, I'll give you an example. There was a lot of publicity around the OBGYN who developed ITP and died like a day or two after the vaccine. Well, if you look at immune thrombocytopenia risk, about 3 to 4 people per 100,000 will develop it in a year. You're vaccinating millions of people now. So some of these associations of people having things and it occurring is just going to happen.
Right.
And I think that's some of what we're seeing. And we're also seeing that, especially with some of the frail elderly, just having a vaccine and even getting out and going to the effort. Yeah, there's some things that I think that we're seeing that are kind of murking the water a little bit, but I think the reality is these vaccines are extremely safe, and you always have to look at what's your risk of severe disease, hospitalization, and death from COVID. And it just doesn't stack up in terms of risk.
Exactly. Yeah. Thank you. Tom Bonig asks several questions, wondering why the government, the FDA, will not advocate therapeutics, HCQ, ivermectin, and others.
I can't speak for the FDA. I think what they're trying to do is look at the preponderance of evidence around that. There's been a lot of papers out of, especially South America, I think Central America, on ivermectin use. There's been some recently on that, some control, some of the randomized controlled trials that weren't that favorable, to be honest, for ivermectin. And then the hydroxychloroquine, the same thing. I think we have to look at the evidence and say where it's pointing to. I mean, even early on, I think hydroxychloroquine looked like in the lab it might have some efficacy, but then if you're not getting enough of it at the cellular level to make a difference, it proved problematic. So I think we just have to be scientific.
Exactly. Yeah. In the beginning, it looks like, I think we're so we so much want to have a wonder drug that initially, when some of the like hydroxychloroquine and ivermectin come out, they look very promising, and but some of the data is controversial. So we just have to, like you said, look at each therapeutic on a case-by-case scientific perspective. So
I think the evidence for actually vitamin D levels is much better, even though you see controversy there. I think the preponderance of the evidence is saying, hey, if you can get those vitamin D levels up to the 40 to 60 range, it has a lot of immune benefits. And so I think there are some things that we can do to help and prevent. And
we've even at our local hospital here, that is one of the we administer vitamin D. So I think it is beneficial as well. He also asked, why does it seem that PCR testing is so inaccurate? He said, i.e., why are there many false positives?
Oh, wow. That's a good question. And I didn't have time to go into that because I had some slides for another presentation on that. But if you look at it, the PCR tests are actually very accurate if you get enough of the viral load. So they all have they all have limits of detection around viral load, and they vary. So if you don't have enough of a viral load in your system, that when you do that swab, that you can then be amplified enough that can be amplified and have a test, you're going to get a false positive test. I mean, a false negative test.
Yeah.
But you have rare false positives. Most of them are false negatives.
Right. All right. And we're just about out of time. We've got more questions we probably have time for, but I'm going to ask a couple more. Bob Carter says, is there any evidence yet of disease severity attenuation with multiple generations of virus transmission?
Now that question was, is there, I didn't quite get it.
So basically he's saying, as in many outbreaks, how, like, for example, with Ebola, after you've had multiple generations of viral transmission, there tends to be attenuation of clinical severity. And he says, have we seen that yet with COVID?
That's a good question. I think that goes to the variants and mutations. And so, I'm sure there's probably some mutations, we're more concerned about those that are making it more transmissible or making it more, sticky or more severe disease. But obviously there's probably just as many that might be tending to go the other way. But again, natural selection tends not to do that. I think a lot of that attenuation you see too is sort of the immunity we build against things.
All right. I'm going to ask you one other quick question. We're just about out of time. HR Accounting says, will the booster down the road, maybe in a year, need to be the same type of vaccine as the original dose you got? So I think what she's saying, if you started off with Moderna, do you have to get a Moderna booster?
I'm going to take a thing and say probably not. We'll see what the recommendations come out, but I don't see why you'd have to do that at that point. But
Okay.
we'll see.
Okay. All right. Very good. Okay. A 15-second answer. Richard Quimby says, I am recovering from what seemed to be a mild case of COVID in mid-January. What would cause after what I thought was being over and back to work, all of a sudden major breathing problems?
Wow, that goes into a lot of the long COVID issues. And so, COVID really is hitting multiple systems, not only lungs, but heart and other things. So we're just beginning to learn about sort of the longer term effects of COVID. I think that's all I can answer in 15 seconds.
Yeah. Dr. McGinnis, thank you so much. That was very comprehensive and excellent presentation on kind of what's going on in terms of vaccine and the mutations. That was, again, great clarity, and we really appreciate that. Just to our listening audience, remember that CME credit is available for this session. The form and instructions will be in your email, which you will receive a follow-up email very shortly. Also, just encourage you guys, please tell your colleagues to join the forum. It's health.samaritanspurse.org. And our next presentation will be Wednesday, April 21st. It'll be none other than Ken Isaacs, who is our Vice President of Programs and Government Relations here at Samaritans Purse. That should prove to be a great presentation. So you guys, thanks so much. Be safe and God bless.
God bless. Thanks.